In this video, we'll be talking about active transport through the use of membrane proteins. So let's start by defining active transport. An active transport is a process that requires the use of energy. And of course, the most common energy source is ATP in the body. Now, why would I use active transport? Because think about it, the body does not want to expend energy if it doesn't have to. But active transport we see showing up in places where I need to move things, especially against their concentration gradient. You know, things usually diffuse from high concentration to low. But there's some cases where I need to move stuff from a low concentration area to a high, and active transport is really the common way that this happens. Now we'll see under active transport there's two different ways, and both of these use plasma membrane proteins, and sometimes they're called pumps. And the two types that we can have a look at are primary and secondary active transport. Let me just give you the quick so what. Is that with primary active transport, that protein pump is going to be moving stuff while expending energy. Secondary active transport is a process where it basically rides the coattails of the first process, is that there is a primary process that happens, and then there's a follow-up process which uses the conditions largely set up by the primary active transport to allow things to happen. Now we'll get into those details. So let's start with some definitions, and that is talking about things like uniport, symport, and antiport. So let me draw. This is a cell membrane right here. Now, a uniport, what it does, it's the transport of a single substance through a membrane in one direction. It could be either into the cell or out of the cell. So uniport, one thing moves across the cell membrane. As I said, it could be either into the cell or out of the cell. It depends on what uniport system you're looking at, what protein, active transport protein you're looking at. But uniport just means one item moved in one direction across a cell membrane. The second one is a symport. And symport is symport, two or more substances moving through the membrane. Now here's a key word, in the same direction. And it could be into the cell. These two or three things are moving into the cell together, or they're moving out of the cell together. But everything's going in the same direction. All right, and let's look at antiport. So let's actually just draw that. So, for symport, it would be, two or three different things that are crossing at the same time. Antiport, let me just change colors here. Antiport is a transport of two or more substances. Now here's the key word, in an opposite direction through the cell membrane. So you can see that we have a difference between symport. Symport, two or more things traveling in the same direction across a membrane. Antiport, two or more substances, they're each going in a different direction. So let's draw that out. So in a antiport, one thing would be going in one direction, and the same membrane protein is carrying something at the opposite direction. So these are very useful terms because we'll be using them on a regular basis, talking about uniports, symports, antiports. So we should be familiar with this terminology. Okay, now that we've talked about the three types of pumps in the plasma membrane, because you're either a uniport, a symport, or an antiport, let's start looking at the concept of primary active transport. And one of the best examples of that is the sodium-potassium pump. And of course, with a name like that, it's not great surprise what you will basically transport. It's now this is not the only example, but it's probably the basic example in the body. So this is a pump. So it's a protein that's going to use ATP to transport both sodium and potassium together. Okay? And of course, this is going to be very important when we start talking about neurons and action potentials and muscle cells, and setting up basically a charge difference between the inside and the outside of cell membranes. Now, just a little FYI, we'll talk more about the sodium-potassium pump down the road, but the pump helps to maintain a steep concentration gradients by transporting let's have a look, 3 sodium ions out and 2 potassium into the cell. And in both cases, this is against their concentration gradients. And it uses an ATP molecule every time I run this pump. Now, let's stop and consider this. Let's have a look. So here it is. So here is our pump protein here. Remember, it's going to have ATP. And what happens is in this case, I'm trying to move sodium out of the cell. And you can see that sodium concentration outside the cell is much higher than inside. So I'm going against the concentration gradient. That's why I'm using a pump and expending ATP. So you can see the protein is in the open configuration for sodium. Sodium comes and binds. And when sodium combines, then what happens is the pump or the protein is going to change shapes or conformation. And you can see it's now changed shapes, that up here, the open end was inside the cell, and now the part of the pump facing the outside world is now open, and the sodium can be dropped off. But remember, this required an ATP to lose a phosphate, and of course the ATP now becomes ADP, but that liberates energy for the pump to work. Now, the pump, now that it's open to the outside world, will allow potassium to come in. Now you notice that sodium sits here and potassium sits here, so they don't sit on the exact same seats in this pump. And you can see once again, potassium concentrations are higher inside the cell than outside. But this picks up potassium, and the pump will return to its original shape. So the pump releases the 3 sodiums in the extracellular fluid. It binds 2 potassiums. And then what happens is the pump is going to change shape, and it's going to return to its original shape. And when it does, it allows for the potassium to exit. So the potassium detaches, the pump changes back to its original shape, and now it's ready to go again and load sodium onto itself. And then, of course, you use ATP, and then we go through the whole process. So this process just goes around and around and around. Now remember, these pump proteins don't get used up with one reaction, right? That they can go over and over. And almost every cell of your body is now running this sodium-potassium pump to maintain this gradient difference. Okay, so that's a great example of active transport. Now let's talk about secondary active transport. A lot of people struggle with this concept, but what secondary active transport is that in secondary active transport, we use ATP, and here's the key word. I'm going to change colors even for it. We use ATP indirectly, all right, to fuel pump transport via the pump. Now, let me clear this off. So ATP is used to create and maintain a concentration gradient of one substance. And then what happens is, once I've created that gradient, there'll be another process that will take advantage of that gradient in order to transport things. So moving one substance across the plasma membrane down its concentration gradient provides energy to move another one against its concentration gradient. And an example of this would be, and I'm going to back up here for a second. The example we're going to use is right over here. Sorry for hopping back and forth. We're going to look at the sodium glucose transport mechanism. Okay. Now what happens is I can use something like the sodium potassium pump, right? Every time I run the sodium potassium pump, I'm going to put more sodium out here. Okay, so I spent energy using the sodium potassium pump, which is active transport. So I've set up the condition that there is a lot of sodium here. Did I use energy setting up that condition? Yes, I did. And this was primary active transport that set that up. Right? Sodium potassium pump uses it. Now that all these sodiums are out here, let's keep that in mind, that I spent energy to put all the sodium out here. Let's look what can happen. Now that all this sodium is out here, and remember how to get there? It got there through active transport. But now that it's out here, I can then use this sodium concentration to pull stuff back into the cell. So in this case, there is a carrier. You notice that you don't see ATP listed here, because this does not need ATP. So the sodium that I put out here by active transport allows a lot of sodium to be out here to basically hook up with one of these pumps. Okay, this is the sodium glucose pump. It allows glucose to be bound onto it, and then the combination of both these, remember this is a SIM port, because two things are moving, it then causes the glucose. So what have I done? Let's just review quickly. I used energy running the sodium potassium pump. I put a lot of sodium out here. So this was the active transport thing. The active transport has set up a condition, which is a lot of sodium out here. And since a lot of sodium is now out here, this allows a pump to run. Now I'm not using ATP directly for this one because I used ATP to put this out here. But since there's a lot of sodium out here, this allows it to bind, sodium to bind to this carrier, allows glucose, and then I allow the glucose to come in and the sodium to come in. So no ATP is used in this phase. So I call this the secondary active transport. And just to remind ourselves, active transport burnt up energy to set up the conditions of high sodium concentration. Once the sodium concentration is high, that allows this mechanism to run. And why is it running? It's really running to move glucose across here. So that's secondary active transport. All right. And of course, we can run pumps, like we just saw the sodium potassium pump. We can do that to set up basically gradients of ionic charges, ion transport, right? And we can have a charge separation that exists across the plasma membrane. And of course, you've seen from previous courses that we see this occur in neurons when they're at their resting membrane potential, which leads to action potentials. So this allows us, by running pumps like this, I can create a zone. Now, the zone isn't really super far away from the membrane. We're usually talking about that zone really close to the membrane, where there's a net increase of positive charges in the extracellular fluid side, and more negative charges on the inside of the cell, or the cytosol side. And of course, we'll come back and talk about that in terms of neurons and how they set up their action potentials. But a lot of this is driven by things such as the sodium potassium pump. Now remember, the sodium potassium pump, just to remind ourselves, every time I run the pump, I pump out 3 sodium and I pump in 2 potassium. Now, 2 potassium. So every time I run the pump, I get a net gain of 1 positive on the outside and a deficit of a negative. And that helps. That's one of the, not the only thing, that's one of the things that helps to set up this gradient that we see here. And of course, we often refer to this as the electrical gradient that we set up. And this leads to a membrane potential, which is the electrical potential across the plasma membrane. And then we can also get into the concept of a resting membrane potential. And this is the membrane potential when a cell is at rest. And of course, this is the best example of this would be resting neurons. Now, just a little heads up. We measure this in millivolts, right? Is that it looks like there's a huge positive charge out here and a huge negative. The difference is actually very slight. So we couldn't measure it in volts. We measure it in millivolts to make the numbers, much more sort of easier to understand. And when we look at it, we see the value is expressed as being negative, which means inside the cell is more negative than the surrounding CSF. Now, these are going to be important concepts when we come back and start talking about neurons, the concept of membrane potential and resting membrane potential. Okay, so both of those will be important. The concept of millivolts and why the inside we write as negative. Okay, we're going to leave this video here, and the next one we'll come back and start talking about how we use vesicles to move big things in or out of a cell.